All right, hello everyone. All right, thanks for coming to the Thursday Moss Landing seminar. So it's my pleasure to introduce our seminar speaker today, and that's Dr. Erik Franklin from the University of Hawaii at Manoa, where he is an associate research professor. So I'll tell you a little bit about Erik's background. So he received his bachelor's degree from the University of California at San Diego, and then he got his master's at the University of Miami, where he conducted a habitat-based assessment of the marine aquarium fishery in Florida, focusing on angelfish. He then went on to do his PhD there at the University of Hawaii, and he did his thesis work where he was creating predictive spatial models of coral species distributions in the Hawaiian Islands. After that, he was very lucky to get to stay around in Hawaii and currently move into this up the ranks through this position as an associate research professor. And his lab is, or one of the lab areas that he works is based out of the Coconut Island lab. So if anyone's been there, it's in Kaneohe Bay. And so he's one of the few people that gets to say that he gets to take a boat to work every day, right? Although it's probably the shortest boat ride on the planet. And his research involves, a combination of field work, laboratory studies and computer modeling. Erik works in both shallow coral reef environments and deeper mesophotic reefs. He uses a combination of open circuit scuba and rebreathers. And his research interests are very diverse. So I was looking at his CV and it's kind of like me, he just loves to do all sorts of different things. And so he has a lot of interest in terms of doing ecological surveys and assessments, studying life history variability and reproduction in fishes, doing fishery stock assessments, MPA evaluations and assessments, right, population and species distribution modeling, and evaluating the impact of climate change on coral reefs. He works on a taxonomically really diverse set of organisms, including corals, invertebrates, fishes, and cetaceans, and has done work in Hawaii and locations all throughout the Pacific, the tropical Pacific. All right, he's also published on a lot of important socioeconomic issues. And one that I really found was pretty cool was a paper kind of forecasting the energy intensive effects of mining Bitcoin and how that can contribute to global climate change. Right, my former student now, June Shresso, was one of Erik's, worked with one of Erik's graduate students before she came to Moss Landing, helping to do some study of herbivory on coral reefs. And I know from talking to her that she really enjoyed working with Erik in his lab. And so we're really excited to have him speak to us today. And so just to remind everyone quickly, so yeah, please keep yourself muted during the talk and don't try to share your screen or anything. After the talk is over, you'll have an opportunity to ask questions live to the speaker. And at that point, you can use the raise hand feature, which is under the reactions tab at the bottom of your Zoom window. Click on the raise hand feature and then we'll call on you and we'll ask you to unmute yourself and turn your video on so Erik can see you. And then you can ask some questions. After the question session, Erik's really excited to stay around and talk more informally with graduate students. So please stick around for kind of the virtual happy hour and chat with him some more informally. And so with that, yeah, Erik's going to speak to us about geospatial approaches to tropical fish ecology and management.
Great, thanks, Scott. I really appreciate that warm introduction. And as we were saying before, I'm sad that I'm not visiting you in person, folks, but as if you're like the rest of us, we're in a bathrobe at home, broadcasting in and enjoying science in the afternoon, at least there, and for me, the early afternoon. Okay, let's get on with it. So a little big picture history here. I like to start when I talk about geospatial work with a Stommel diagram. And a lot of today's talk will, I think, incorporate elements that I kind of also call public service announcements for early career folks. If you don't know the name Henry Stommel, after the talk, look him up, because most of what we understand about physical oceanography, I would say second to someone like Walter Munk, is probably from Henry Stommel. And he came up with this idea of a Stommel diagram, which is basically a way to look across space and time and identify how biophysical phenomena relate to both of those. And then also overlaying what's our ability observationally to try and understand or capture some of these phenomena. And I'm not going to go into the detail of the figure itself, but what I want it to do is really demonstrate this idea that scale matters in our science, especially when you talk about ecological sciences, understanding the space, the time, or spatiotemporal scales that we use is quite critical to the work that we do. And for those folks that want to learn more about these Stommel diagrams, there's a great paper by Vance and Dole that I suggest that you go for. So this space-time issue, it really leads to these questions of where. A lot of my science in ecological fisheries, whatever it is, I'm often thinking location. Where's that reef? Where's that school of fish? You know, certain unique species, big fishes, little fishes. Where's the juvenile stage? Where's the adult stage occur? And you know, this is obviously also my, like, here's Hawaii and how beautiful it is, and you should come visit. Where's that turtle? You know, what kind of habitats do they utilize? And that question where has driven a lot of the science that I do. I found that to answer that where question, it requires geospatial data tools and methods. And so we're all talking the same language. When I describe these things, what do I mean? So the first really big technological advancement is global positioning system. And this is this constellation of satellites that we all now take for granted. If you're born in the 90s, you're like, oh yeah, GPS, it's been around forever. It's like, I've got it in every device I have. For those of us that have a little gray in our beard, we use things like LORAN. And there was a time before GPS when you'd go, look at that mark on land and look at that mark on land and let's go find our spots. But this constellation now, which has been operational for a couple decades, really opens up the ability to perform geospatial work. And so geospatial data can be almost anything, right? We're talking biotic, abiotic, observational information, experimental information, simulation or model output, but it's all got that specific locational attribute. And often what that means is it's got coordinates in some geographic projection. And a projection is just a way to take a flat map and project it onto a curved surface like the Earth and be able to work with that information. How is data typically collected? You know, when we talk large scale, say macroecological scales, often remote sensing is the way that information is collected. And so it can be sea or shipborne, airborne, whether that's a plane or a drone, satellite sensors that collect all number of data, including imagery of these biotic and abiotic data types. And once we have this information, what are we typically doing with it? Well, we're usually plugging it into a GIS. And when I say GIS, you know, it's this software that allows you to merge data management analysis with geoprocessing functions and cartographic visualization. So you've got a geographic way to look at where your information is and understand relationships. If you've heard of ArcGIS, that's the most common. QGIS is an open source version. But I kind of broadly, you know, GIS can also be just mapping with any software tools, can be R, can be whatever you use. And then I think the last big component of this is now internet mapping. This is really something that's only, you know, barely a couple decades old. And these are the network software and server components that provide remote users an ability to interact with geospatial data. And again, as I said, we take this stuff for granted now. Every one of us has a smartphone with a map, and we can do with it and, you know, say, I want to go here, I want to go there. This stuff is so novel for folks that maybe started their career, you know, say in the 90s, that I want you to appreciate how much it opens up. This is an avenue for research. And what are potential topics that we'd be covering when we talk about this? Well, at least from the ecological side, and I'll build from the ecology to the management, we've got things like habitat utilization. So this is identifying associations between biological populations and habitat characteristics. You could determine areas of, say, high abundance or biomass, diversity. You could look at ontogenetic habitat associations. That also then feeds into things like species distribution modeling. And this is a process where you're fitting observational data or any kind of data about species occurrences to models. And those can be, say, regression-based, machine learning-based. And then you will predict the species distribution and abundance or occurrence from this set of biotic, environmental, or even anthropogenic covariates that you use in the models. You also could look at seascape ecology or marine landscape ecology, depending upon how much of a hardliner you are in that niche. And this determines the influence of things like seascape patch or habitat structure, adjacency or distance on different biological components of these species you're interested in. And all of that feeds into this broader ecosystem scale modeling and simulation, where you can use coupled biophysical and ecological dynamics models to better understand the biological community, its structure, function, and trends. And for the management side, a lot of that ecological data then can feed into things like fisheries independent surveys. So this provides observational data and environmental information that then can be used as a proxy for things like relative abundance of a fish stock. And one of the kind of gold standards is this unbiased collection of fisheries independent data, which can hopefully reflect what the dynamics of that population are when it's being fished through time. Another approach is for evaluating the effectiveness of spatial management areas. And this can be inside and outside MPAs, whether it's a backy design or just a space for time replacement, like I mentioned, and even things like the distance to boundaries. What's the effect as you move away from an MPA or towards it? And then within the fisheries realm, essential fish habitat is something that the Magnuson-Stevens Conservation Management Act requires for managed species. And this is delineation of habitats used for things like survival, growth, and reproduction of these fishes. And then finally, the ecosystem-based fisheries management approaches kind of ties all these together in a geospatial or geographically explicit framework to understand species interactions and how fisheries basically interact within the natural systems. And so that's a lot, right? I think like Scott said, Scott's got a lot of interests. And so what do you call all this stuff? And kind of the umbrella that I use often is called Ecoinformatics. And Ecoinformatics is this framework that enables scientists to generate new knowledge. And it's using tools for collecting, discovering, managing, integrating, analyzing, the whole menu here, visualize and preserve the data that you use for your work. And this ecoinformatics, you may have heard of like informatics of ecology or ecological informatics. I kind of like this ecosystem informatics as a definition, where it's much broader. It's kind of everything under the sun, the biological, environmental, and anthropogenic elements that go into it. And I really feel like this structure, it provides kind of a pathway for ecology to become a data-intensive science, as well as what I would call operationalizing ecology. Because when I communicate on the fisheries side, I'm often talking about how do we take research, make it into a product that fisheries can use, and in an operational sense of year to year, using that information to basically then inform management decisions. And if we look at a Venn diagram of what is ecoinformatics, it's really this intersection of ecology, computer science, and math. And there's kind of the different sub-disciplines that go within, but at least for my own research, I would say I'm a trained quantitative marine ecologist and a fisheries scientist, but I dabble and utilize tools in mathematics and computer science. And so to structure our talk, the rest of our talk, I'm going to give this, there was a nice figure about 10 years ago in Tree that talked about data life cycles. And for geospatial data life cycle, I'm going to use this to kind of walk through some of the work that we do, and also dabble some little PSAs for early career folks and maybe late career, mid-career folks too. So typical life cycle, right? We plan a study, we go out, collect some data, we do some QA, QC on it and assure it's what we think it is and it's defendable. We do some descriptive work, and often that just means metadata, or the data about our data, so that we know what we're doing. We've got a good handle on the methodology, and if it needs to be repeated, we can go back and look at that. And then we analyze that data, and typically then we publish, hopefully. That's traditional, right? So this next stage, at least with these new tools that ecoinformatics has brought about, is adding these additional steps of, once you've described that data, then you preserve it. That means pushing it to an archive. And that can be for your benefit or for the broader scientific community. And then that allows you either other folks to discover your work, or even often for you to rediscover your work, as I've found in my career. And then you can integrate that and integrate it into the analysis for whatever your studies are. And that could be pulling other biotic or abiotic information to really kind of make your study more robust and get at some information that you're not able to collect yourself. And so as we work our way around that cycle, that first step is the plan step. And this is kind of a plug for early career folks. And this is something new, I would say, in the last decade or so, where most federal agencies in the US, when you submit a proposal, they're going to ask you to submit a data management plan. And 10 years ago, most of us that got that request were like, I have no idea what you're talking about. And so fortunately, there's a number of tools that have come out. One of them is this DMP tool that allows you to structure, okay, what is my study going to consist of? What information am I going to collect? But then also, how am I going to manage it? Where am I going to archive it? You know, what's the accuracy of that data? Who can use it? You know, all those questions. So this is a great place if you're not familiar with these concepts to go and utilize this tool if you're not already aware of it. And so moving around our life cycle here, once we've got a plan, we're going to go out and collect data. And when I talk about geospatial data in the sort of tropical environments I'm in, you know, often we're fortunate, we've got really good water clarity. We can use things like aerial or satellite imagery. And so this is an example from Pearl and Hermes Atoll in the northwestern Hawaiian Islands, where you can get a satellite image and then use some statistical methods to come out with things like a pseudobathy or a bathymetric map to give you an idea of what are the depths of those areas that you can see. And then you can also use that similar approaches to then map what type of habitat do we see? Is this sand? Is it reef? Is it land? Is it something in between? And this now gives us kind of the finite domain of what our study is, how much habitat area there is, how it's distributed, you know, what's the adjacency to other regions that can help us to ask some of these geospatial questions. And if we zoom in on that benthic habitat, it's also now a means to allow you to structure things for a stratified survey, for instance. When we talk again about that fisheries independent data, each of these grid cells now becomes a potential random draw from your survey design. And this is a nice way to kind of keep bias out of your selection of sites. And it then feeds into some of the management needs too, when you've got these population estimates that can come out of these surveys. So that was a little broader scale. Some fine scale work that we've done too is utilizing drones for habitat mapping within Kaneohe Bay. And this figure on the left, this is an aerial photo of Kaneohe Bay. And I don't know if you guys can see okay. So this little island just down here, that's, as Scott mentioned, that's HIMB, that's Coconut Island where the lab is based. So we have ready access to this whole bay. And this was a study, it was actually to look at coral bleaching, but you can get the idea that it can also be utilized for what I would say high resolution habitat mapping for, say, fish utilization, for coral abundance population mapping. But this allowed us, using just commercial off-the-shelf drones and some software, Agisoft, which is basically like a mosaic stitching software, to create, this is an amazing, I mean, it's so much work, and this picture just doesn't do it justice because it's actually a fairly large patch reef, but it's amazing detail. And you see a little bit of an inset there, that black rectangle on the right side image. This here is actually this image. And you can see the resolution where now you can actually pick out individual coral heads. This is a huge leap forward in terms of the type of imagery that's available for us. And it kind of opens up just how, you know, I mean, seeing this in my mind just opened up all these different questions. And I hope it does the same for you. But this is some work that I did with a student a few years ago. And then when we're collecting data about fish, typically it's an observation by a diver. And, you know, it's the usual transect, or it's a stationary point count. We're writing stuff on a slate, or we've also been experimenting with diver held stereo video cams, or we've also deployed stationary stereo video cameras at the bottom. And so I think one of what I'd like to do is share with you kind of a future of where I think a lot of this work is going now, is using deep learning AI to semi-automate a fish count process. So what you're seeing is video, and this is a collaboration with some colleagues out of the University of Philippines that developed the computer science algorithms here, that basically you can train them to identify the fish. And then with the stereo video, they know the size of these objects. So as you see a box come into the screen, it's saying, okay, there's something there. And now I'm going to mine all of what I've learned about fishes to try and identify that to species and give a size. And we've got relationships between size and weight. So you can also estimate biomass. And so this gives you, you know, things like max N for species, different species biomasses that you find, a length frequency, you know, a histogram for these. But I guess my message here is automation is another step that's going to really push ecology into this kind of operational science. The reason why oceanography can, I think, jump ahead and collect so much data is because a lot of their processes are automated. So the robots are coming for our jobs. But anyway, this is a pretty exciting thing that we're working on now. Okay. And the video worked, yay. Okay, continuing to move along, this describe step. So describe is really the metadata step, but I want to do another PSA here, public service announcement. Information entropy is real. Entropy dominates the universe, okay? And so this figure, we've got information content, which is at its highest at the time of publication. And as time progresses, we start to lose information. And this is just in our own head. Maybe we can only recall specific details of something. A little more time passes, we can just go general details to the extent that you go, yeah, I remember I did some surveys a few years ago, but I couldn't tell you a thing what we saw or did. And then if you're unfortunate, an accident happens, that's all lost. And then as you slide into that retirement or career change, you sort of drop off that, that severe, let's say, now I'm going to be a watercolor painter, and just all of that information is lost. And then we die. And even after your death, maybe you've got students or colleagues that are around that might remember something about it. But the whole point of this figure is this information, all that you're collecting in your scientific career, unless you're documenting it, creating metadata, and archiving it, it's as if it never happened. You can publish a paper, but if you want to share that information and have it reused, you need to basically do these steps to help to describe and preserve it. How do we do that? Metadata. I'm not going to get into the nuts and bolts of that ugly thing on the right side here, but for those of us that have been doing metadata for a while, there used to be a green book. There still is. It's a PDF. You have to go through this thing and follow all the instructions on how to do it. But basically, there's international standards for how to write metadata, and there's different schemas. Some of those are CSDGM or Darwin Core if you have a taxonomic focus, or ecological metadata for a lot of the things we do. And these are all governed in the US by this National Spatial Data Infrastructure. And there are some tools out there. If you, after this talk, you're like, you know what, I'm collecting data and I'm not doing metadata, here's some tools. USGS has this online metadata editor, or OME. Morpho and Metacat is an NSF product. Metavist is a forestry service. And then ArcGIS will also allow you to utilize, create, and manipulate metadata. So again, metadata, metadata, metadata. Okay, PSA, that's PSA number 2. Here's PSA number 3. Once you've got your data and your metadata, please preserve it, store it, archive it. And at least for marine scientists, most likely that means it's going to the National Center for Environmental Information. That's housed by NOAA. And typically, at least for marine biology, it goes in the Coast, Oceans, and Geophysics wing of that division. There are other options, and we've used these too. Data Dryad is an open source repository. CDataNet in the EU for the European Union. NSF has BCO DMO. And these are just ways for you to identify, discover, reuse data that's already been collected, because that's an investment that's been made, especially if it's federally mandated and federally funded research. It should be discoverable. And this is a fairly dated table, but it's one that I could find that would identify some existing collections, and often they're either a clearinghouse or they're organized around taxonomy. So we've got the Ocean Biogeographic Information System for ours, but you can see kind of examples of what's out there, what at least was 10 years ago. And so continuing to kind of move around our life cycle here, we've got our information, we've described it, we've preserved it. Now we're going to analyze it a bit. So to kind of give you a little detail, Scott introduced this a bit, but here's some work that we did that involves species distribution modeling. It's really a basic question. How much coral is there? How does this influence, habitats and the availability for fish? How does it influence, what's the bleaching effects? Like, if you don't know how much is there, you really don't know how things change. And so one of the activities that we were engaged in was taking about 10 years of coral surveys. And these were data we collected, data federal agencies collected, coming up with a synthetic database. And you see the sort of sweep of the main 8 Hawaiian Islands, these guys, and around them are actually black dots. Each of those black dots represents a location of a survey point. And so we were able to pull data out of all of those locations, and you can see the spatial coverage is quite good. We were also able to pull and integrate data on things like waves from model output, depths from bathymetric mapping activities, using that bathymetric data to derive things like slope or rugosity, and also calculating the downwell irradiance, because corals are symbiotic with zooxanthellae that photosynthesizes. So they also are, the irradiance is important. And we used a machine learning approach called boosted regression trees. This is just a way for you to basically fit a model to data that helps you predict where and how much coral is there. And basic tools, and this is kind of pretty common for most of the work we do, are R, ArcGIS, and Python. And this lower level of figures are some of these environmental covariates that you'll see. So from the left to the right, it's sand habitat. So the more yellow it is, the more sand, the browner it is, the more hard bottom. And then we've got max and min wave energies. One of the largest predictors of coral habitat distribution in the Hawaiian Islands is wave energy, as you would imagine. Anybody who's a surfer, who's seen any of the surfing competitions, knows we have a phenomenal amount of wave energy, almost matched by Moss Landing. And downwell irradiance there is the last one. And so what do we get out of these? We've got our observations, we fit our models to that data, we have these models now, and then we also have these spatially explicit sets of environmental covariates. And we can use the models then to predict how much coral would there be in these locations where we haven't actually observed yet. And so these are, you would say they're a map. They're actually spatial hypotheses, but they're hypotheses that have some accuracy and some validation. So you have some faith that they actually reflect the system that you're trying to observe. And so this is just an example of a meandrina. This is this hardy little coral on the left that likes to be in areas of high wave energy and just high movement. And these are the kind of tips of Oahu, which is the map of the island that we're seeing. So the hotter colors, it's more frequent there. And then we've got a habitat builder, this Posilopera lobata, or sorry, let's get rid of that, Parietes lobata, which we find is pretty dominant on this windward or east side of the island. And this is a primary framework builder in Hawaii. And then another species, Parietes compressa. This is this finger coral. It's a bit more fragile. There's a little bit more complexity to it, and it's more dominant in protected or sheltered lagoonal habitats. And you can see on this eastern side of the island, this is Oahu, that's Kaneohe Bay. And so this finger coral is really dominant within these patch reef areas where you don't get that same wave energy within there. And so this is a way now we can not only have predictions about them, but these can feed into other activities. So we can do it for corals, we can also do it for fish. Here's another study where we looked at what are called the Deep Seven, and this is a complex of deep water snappers and a grouper in Hawaii that are not necessarily as sort of economically important as tuna or some of the pelagics, but they also have a very high cultural value in Hawaii. And using very similar approaches, we were able to map out what's the predicted spatial occurrence of these. And these are the kind of things that, at least for managed fish species, can be utilized to inform a delineation of essential fish habitat, or EFH. And you can kind of see, again, hot colors mean much more prevalent. So some of these species are right on the margin of the drop-off. Some of them utilize, much shallower or a much broader region. And you can really winnow in on, what specific habitat types are these species interested in. And this is work that I did with another of my students, Zach Oyafuso. And part of, when I say the model, the outputs for the model kind of look like this, where you've got, these are just the fits to the data for the environmental covariates. And I've pulled out, this was from the paper, some of the more important covariates that were used to predict. And we've got three different kinds of models that we were comparing: a GLM, or generalized linear model, a GAM, generalized additive model, or a BRT, again, the boosted regression trees. And you can kind of see they vary in their flexibility of how they can actually fit to data, where a linear model is obviously aligned, a GAM has some flexibility to fit, and a BRT is extremely flexible on how it can fit. And you can see that allows, an evaluation also of modeling approaches. And you can then look at certain things like, say, the backscatter column, where, for the most part, when you go from 0 to 225, that higher value means it's more hard bottom, lower value, more sand. And so you can see these fish like hard bottom. And pretty much whether you've got a GLM, a GAM, or a BRT, they're really saying, you know, as you increase the hard bottom, you're going to have a higher predicted occurrence. Some of the other variables aren't as clear. So if we look at bathymetry, you know, it looks like some of the species actually have a Gaussian or kind of a humped function. And a thing like a linear model can't really pick that up, but it looks like the GAMs and the BRTs are able to pick that up. So comparing these modeling approaches can be a useful step when you're trying to determine these species distribution models and what's a good predictor for their occurrence. So continuing on, this is kind of like the tour through a lot of the work that we do in the lab. Marine landscape ecology, if you're a hardliner, or if you're not, seascape ecology, is this idea of taking, you know, the explicit geographic locations of your habitat or your patches of interest and trying to use those to learn something about biological communities that inhabit them or use them. And so this is up again in the northwestern Hawaiian Islands. This is Midway Atoll of the famed Battle of Midway. And from left to right, we've got benthic habitat maps of varying resolutions where you can see, you know, when you've got a really high resolution map, you can see all the kind of detail all the way to this almost blocky, you know, it looks like, what's the game? Oh, I'm going to forget. It's like there's a blocky video game. Oh, the kids all play it. Anyway, and so it's this you know, this range of resolutions. And then that can influence things like how many actual habitat patches exist at these different range of map resolutions. And you see, obviously, they decline as that map resolution gets more and more coarse, because the patches then become incorporated you know, with each other as that resolution gets coarse. And you can then kind of, when you have these distributions of your habitat, and this is at the higher or highest resolution, you can ask questions about things like, you know, on the left column, it's reef area, and on the right column, it's the distance to the reef crest. And that means you're in the back reef, you're in a sheltered zone. The reef crest is probably the highest energy, wave energy environment in the reef system. And how do these things influence a number of fish species? I could have just put species richness. So how does that influence things like species richness? Or how does that influence things like the average size of all the fish, not just a single species? And we see that there's a slight to almost no relationship between reef area and those two biological variables. You know, it's a little bit of an increase. But a thing like distance to reef crest, as you get farther away from that reef crest, we're finding that species richness went down and the size of those fishes went down. And so there's something about being in that area that, even though it's higher energy and maybe, you know, to us, it's somewhere we would never want to dive, there's really something going on there that leads to bigger fish, more predators, and a higher, you know, the average length size means you've got basically higher trophic level fishes present there. So these are ways to take those marine landscape, you know, basically a distribution of habitats, and start to think about how do those maybe influence the within ecosystem, you know, structure and function of these species and communities. And another way that we've used these geospatial approaches is for MPA effectiveness. You know, MPAs are a spatial management tool for fisheries or conservation. And really, you know, a basic, easy way is kind of this geospatial data is used to develop our stratified random sampling designs. And often it's like a habitat by management. And we can look at then things like, you know, we're able to take an unbiased sample from these environments. Because often I'm kind of a stickler for like, you know, in ecology, we sort of know what the questions are as we're collecting data. And so the best way that I've found to try and, you know, circumvent any inherent bias is to try and get these nice, clear, stratified random sampling designs in place where it's telling us the location to go to. We're not seeing it and picking it. And then you can do things like comparing, this is herbivorous fish biomass inside and outside MPAs or combined for all MPAs. And this is a recent paper that we came out with that showed that at least around Oahu, most of the MPAs aren't doing a super hot job. Hanauma Bay is, the others not so much. But if we combine them all, we find that Hanauma Bay actually has an outsized effect on the island scale assessment, such that it seems like MPAs in general are doing well. And so it's a little bit of a cautionary tale that, you know, make sure you're looking I would say MPA by MPA, MPA versus reference habitats outside when you're trying to make decisions about the effectiveness of those MPAs. How are we doing on time? Okay, good. And then another one is this, the kind of big scale stuff. If we zoom back out and we're interested in ecosystem scale questions that can feed into fisheries management, management strategy evaluation, or just trends in populations, what are the effects of natural anthropogenic drivers, we get into kind of this ecosystem modeling. And so this is some work I did with a student a few years ago, where we have a coupled biophysical models with ecological dynamic models at a local scale for at least these functional groups. And they interact with just the typical, differential equations. And this thing just rolls through time, and they're connected by a transport layer that's a static transport layer after you look at things like, upstream, downstream connectivity for reef sites. And then that allows you to also play games about putting MPAs in. One of the big pushes right now is 30 for 30, which means to try and protect 30% of habitats by 2030. For our study, we didn't actually include 30 because we didn't think that would be feasible. But we did a range for this as 0, 5, 10, and 20% MPAs. And we ran this simulation out 100 years to see, what are the effects of things like coral bleaching, continued fishery pressure, some eutrophication and storms on functional groups like corals, turf algae, herbivorous fishes, and piscivorous fishes. And you can then kind of look at these trends and make some predictions. And that what we're finding is in the near term, having MPAs is a good thing. But as coral bleaching basically diminishes coral reefs to the point of almost extirpation, MPAs do little to protect these habitats. We will still have fish, and especially herbivorous fishes, as long as there's substrate for things like algae, turf algae, macroalgae to grow, but there'll be a diminishment in these communities overall. And so these are kind of games you can play. This is, I would say, an MSE approach to spatial management, but it also, there's a number of questions that you could address with these sort of existing systems. Okay, and kind of the last just point to bring up is there's, I've talked about all these resources and integrating them. At least in our region, I want you to be aware of, there's the Pacific Islands Ocean Observing System. There's these IUS observing systems all around the US. So for your region, I'm sure there's one. But they're great resources where you can actually pull a lot of the kind of data that I've shared with you today. And if you haven't heard of it, I would highly recommend to the ecologists out there, AirDAP as a server that you utilize to grab data. It allows you basically to have a persistent RESTful URL. And so if you're using R or you're using Python where you're pinging on a server and grabbing data, this is a way for you to basically utilize that information as fresh and current as it's currently sitting on the server, so that you're not always having to deal with static information. And then other data out there, I already mentioned the NCEI. If you're looking for other information to inform your studies, that's a great place to go. Also is your archive. Data One is an NSF-funded portal that allows you to search a lot of this. And again, for oceanographic information, or for ocean, biogeographic information, OBIS is really the place to go. Okay, and so in summary, a geospatial approach allows you to address those where questions that you can come up with in fisheries ecology and management. And I think as you've seen me kind of give you the buffet version of the research that we've done in the lab on this topic, managing these data and dealing with them is pretty complex and challenging. And so I suggest if you have that interest in computer science or maths, get trained in it, be proficient in R, Python, whatever tools are out there to help you kind of tackle these sort of problems. Also, ecoinformatics are awesome. This is the framework. This is really provides this foundation for ecology as a data-intensive science. And it provides that framework for that life cycle of the geospatial data that we walked through. And the last PSA, folks, document your data with metadata, archive it, share it, get it out there. It's kind of historically hard for ecologists to share because we put so much blood, sweat, and tears into like, this one table took me years, but get it out there and share it so it persists beyond you and your career. And with that, I'll say mahalo, which means thank you, and any questions. And there's my contact. If I'm going to hang around for, I guess there's a virtual Pahana or virtual online get together. I'll be around for that if anybody wants to talk, but otherwise you can reach me at that email address. And thank you very much, everybody. Really enjoyed it.
Awesome. Thanks a lot, Eric. There were some really great messages there for our graduate students. I really appreciate that. And yeah, it was really interesting to see just kind of a window into all the different types of research that your lab is doing. It's really fascinating. So yeah, time for questions. Does anyone have a question for Eric? All right, we have a question from Scott Schaefer. Go for it.
Okay, well, it's great to, I don't know if my video is on.
It is.
Turned it on. Okay, perfect. It's really great to hear all the different aspects and your PSAs, and the similarities between what you do and what I do for seabirds is striking. But I guess my point or question that I was going to mention is just, I'm glad you defined ecoinformatics, because my understanding of ecoinformatics was based on how you described it, but I've heard lots of people describe it as, oh, it's molecular ecology and all that, when in fact, this seems to be the more common definition of ecoinformatics. And I think it's a really important field, so much so that my understanding is that some universities are now actually starting to offer advanced degrees kind of in this this field.
Emily, I think Eric is muted. So you got to unmute him again.
There I was going to work on my sign language. Hey, thanks, Scott. I appreciate that comment, question. I guess I completely agree with you, because I think us ecoinformatics practitioners are not doing a great job of getting enough papers out that define and utilize the ecoinformatics term in the way that we do, because in my experience, it does get co-opted into bioinformatics, and bioinformatics is much more the kind of, health science, biomedical approach. But when I say ecoinformatics, people are often like, bioinformatics? What do you mean? And so I think this is a good, platform for sharing this expansive idea, because it is so separately different from what bioinformatics traditionally is. So I will do my best to publish an ecoinformatics paper to help push it forward. But hey, if you're interested, maybe a cooperative opportunity there.
All right, any other questions? Travis has his hand up. You're up, buddy.
Hi, Eric. Thank you for the talk. I also really enjoyed all of those PSAs. I've got mine scribbled down here, so hopefully I'll use them. So my question is a little bit theoretical. You made a point to encourage people to share their data and get it out there, and I totally agree. But I'm wondering, do you think moving forward, as people start to, the data sets are kind of exploding exponentially in size, especially with the addition of more complex imagery, do you think that it's going to be harder for people to share their data as these, file sizes explode and sort of take off?
Yeah, it's a great question, and I think the response that, as soon as you were talking about it, what came to mind is imagery, and imagery or video data, and it's a problem that even if you go and talk to a lot of the generators of this, and everybody collects it now, there still isn't a good answer for how we're going to archive this stuff. I think it's actually, now as we're talking about this, it reminds me, I took a trip to Mbari, and one of the most sophisticated video management systems I've ever seen is in that shop. And they use it, I think, for a lot of their sub work. And so I think that would be a great place if you're ever thinking about looking like, how do we document and kind of keep track of this? But until we have propped up, a large scale repository or way to kind of deal with this, I think it will be challenging to, especially if we're talking unannotated video. But so that's the kind of caveat. I think the flip side of it is, I didn't present it today, but I'm working with another student who's recently using convolutional neural networks to identify marine debris on coastlines. And I think the tools he's getting access to through things like Google and their TensorFlow, I think what we may have are these farms of raw imagery that then we just turn AI loose on it. And then there's this kind of intermediate level of data that can be mined that I think will be a little easier to access, that it's, sort of got a post-processing that says, okay, what are you interested in? And then you can kind of mine those upper layers and then figure out how you want to dive into it. But I do think finding an appropriate system for managing that is probably going to require more collaboration with industry and just finding solutions that are on an amazing scale, because it is almost, we're generating, a simulation that's basically a copy of our world now. So it's getting so huge. It's all about technology, but I think we'll find some solutions that'll help it.
Yeah. Well, yeah. Thank you for that thorough answer. And then hopefully a quick one, if I may. What's the software that you use to analyze that stereo video?
Yeah, it's called Event Measure. So it's a the video that I showed. So that's called Fisheye, and so it's University of Philippines. My collaborators are Pros Naval and Lord David, and it's basically, there's a website now, but you can reach out to them directly. They're interested in expanding to new folks to work with. It's a great tool. I mean, if you want to talk about it more offline, I'll be happy to give some more in-depth descriptions and details on it. But it's exciting stuff.
Yeah, no, it looked cool, and I've kind of dipped my toes into event measures, so that video really, that excited me. Thank you.
Yep.
Yeah, that's great. We have a number of students who've been recently doing some stereo video, and one student who just lowered his hand was Cameron Strickland. And I think you just answered his question because he's been doing 3D stereo video, and so he was probably really interested in that. Oh, he raised his hand again. Cameron, you want to ask a question?
Yeah, hi. Thanks for the talk. Travis's question kind of poked at what I was getting to, but yeah, when can we just sit back with a martini and let the computers do all of our event measure? Like, how far off is that, and what kind of collaboration is that going to take?
Yeah, it's a good question, man. It's hard to put a crystal ball to when the robots will take over, but if you kind of look at, I had some of these, the early slide when I defined what the tools were. You look at something like GPS comes online in '93, Google Maps comes online in 2008. This is not a very long time ago. So, I mean, I would say on the scale of probably decades, where you can essentially have, your fleet of, one of the things we're looking at trying to find a collaborator on is drone swarms, where they communicate with each other, but they're also following certain targets in the environment. And they can be used for things like, let's follow fish around. And basically you just let them loose and they just tell you, what used to be active tracking with, at least in coral environments, is like a guy in a kayak with headphones and like, where'd that fish go? And like, oh, okay, I think I'm here and I'm close. You literally can have this coordinated network of robots doing that work for you. That stuff is, I mean, it's available, at least cutting edge now, in terms of, something that we'll commonly be using. I think decades is the probably proper scale to say, but it's coming.
I was really impressed with the fisheye software, especially thinking about how challenging it must be in a coral reef environment that's so speciose, right? And there's so many different things you have to train it to identify. But it was really impressive just seeing what it could do.
Yeah, thanks.
Rick, you're up.
Okay, well, thank you. Yeah, Eric, I really enjoyed your talk, and I agree with you on most things. One of the biggest questions that I have right now is, how are we going to archive all of these videos and all of the reams of information we have? for future use. And I'm glad to hear you're optimistic about it because it's going to take a dedicated coordination from a lot of people around the world. But my question for you is that, I had a student a couple of years ago who was working with some GIS modeling, and we asked the question that you presented related to the resolution of the data. And, we noticed that our estimate of the abundances of fishes in an area changed by an order of magnitude or more, depending upon what resolution we used on the map. So as you described, if you look at an area, you can look at many small habitat patches forming a mosaic, or at a larger scale, it's one habitat patch. And I've been scratching my head to figure out which is correct, because I think we overestimated the abundance of fishes with the larger scale of analysis. And with the finer scale, I think that we may be overemphasizing the importance of certain types of habitat. Have you, I'm sure you've thought about that, but have you kind of come to some conclusion on that?
Yeah, the one piece that I think also influences that is, mobility of your study species and whatever your methodology is that you've selected, and how that influences what your true effective sampling area is. And we're starting to grapple with this with some of the stationary point count or stationary video count data to try and also, so I'm kind of answering your question tangentially, but by presenting that I think there's also another level in there that influences why you may have biased under or over estimates of the actual true abundance. And I think it is a hard problem, right? In my experience, what we haven't tried to push it out to a really coarse resolution. We're usually on, the order of, should we do, say, a 20 square meter to 100 square meter? But I think it would be an interesting activity, now that you brought it up, to say, okay, if we push this to kilometer scale, what are we losing or how does this affect our biological estimates? I think we're probably spoiled, Rick, in that we have pretty high resolution information. And so we just default to the higher resolution information gives us a better depiction of that environment.
Yeah, I think that, our what we've been trying to do here is trying to figure out how to add stock assessment information on the West Coast. And so you're dealing with some areas that have, 2 meter mapped resolution, and other areas that have 50 meter mapped resolution. And kind of figure out if the goal is to estimate the number of fish in a 100 kilometer stretch of coastline or 300 kilometer stretch of coastline, which might be the appropriate scalar for that. And right now we're just kind of scratching our heads because, as you say, we tend to think that the smaller resolution, finer scale resolution is giving us a better information. But a lot of that I think is dependent upon the movements of the fishes as well. So that's, maybe we can talk offline at some point, and I can get your thoughts more about that.
Yeah, I'd be happy to do that. I do. I mean, you're kind of giving me some thoughts too. We really, the resolution of a lot of, when we're trying to get things like population density estimates or abundance estimates, the resolution is kind of what we call this, the base map scale. And we'll have environmental covariates that have either a higher resolution and sometimes a lower resolution than that, but it really depends on kind of, as you would say, the home range, or, do we think that this particular animal is going to be contained within our sampling method or our field sampling method. And then we basically use that as a bin that we think reflects the biology of that organism as kind of accurately as possible. And that's sort of how we come up with an optimal distribute or an optimal resolution, at least for the analysis. But I'm happy to chat with you more offline. This is the kind of stuff I love and think about. So.
Yeah.
So
So
I've always
thanks.
been really fascinated with the predictive spatial distribution models, right? Especially when you can get up to abundance and biomass estimates. And my question is, based on your experience, I mean, how many samples do you think you really need for the biological data to produce a map that is a close reflection of reality? Like, yeah, how much sampling do you really need, I guess, to do a good job at predicting what it looks like in those unsampled areas?
Yeah, like most things in biology, it depends. You think about the different elements that go into it. It's how diverse is the sort of domain that you're trying to sample. How sort of evenly distributed or variable is the distribution of the target species that you're trying to get a handle on. What are the sort of complexity of the covariates that you think actually influence this distribution? And I would say, as those things add up, as you have more and more strata or more complexity and sort of the things that you think drive it, and more variability in the distribution of these animals, I think that's what sort of increases your sample size. But in terms of an actual rule of thumb, gosh, that's hard to do. I mean, it really depends. You can get, I mean, I've made maps that are fine with, a handful of covariates that you're dealing with and a couple dozen samples, but it's not something that you'd sort of make management decisions based on. So again, it just really depends on the complexity of the environment that you're dealing in.
And have you been able to go back sort of after you've made one of these maps and kind of test some of those unsampled areas to see how well you did, I guess, or do you typically just use only part of the data set to train it and then the other part to sort of interrogate it and see how well you did?
Yeah, we're fortunate here. There's enough activity that we can actually, the kind of gold standard is you train and then you test with an independent data set. Usually you don't have an independent data set, so you end up splitting the data set you do have into train and test. But we have basically, sort of full programs that are independent of each other that we're able to kind of compare. And I mean, obviously the test models usually don't fit as well as the trained models because it's less data go into them sometimes. But they're typically comparable. But we find, we find that I think the approach, I don't know, how can I phrase it? I haven't really gone, I mean, actually some of these questions are good. There's some good simulation work that's been published that looks at like, okay, we have a known distribution. And I guess my answer is going to kind of come back to, it's hard to compare these things when you've gone back because you don't actually know the true answer. You're still estimating. And really the way to do it is like in a simulation environment where you actually know what that simulated ecosystem contains. And then you can kind of play these games of like, okay, if we had these different level samples, you're always going to have noise when you're trying to play that game in a real environment.
Yeah. I think Rick has one more question, and then maybe we'll transition to the virtual happy hour.
Yeah, I'd like to follow up on that last question you answered. And my experience is that when we go back to test our species distribution models, that we don't have very good success in reproducing it. And again, that's part of the issue there is a temporal variation. Mm-hmm. So you don't know what's real and what's not real when you're testing your models. So it would be, I think that's another area that I think needs some work in the species distribution modeling world, is to figure out, and as you say, maybe model what the real landscape looks like with fishes and as they're milling about and interacting with your sampling tool. And maybe the answer is to sample an area for 3 or 4 or 5 different times of the year or different years and see if you can make a a map that incorporates that variability. But I think that's something that needs work as well.
And that was going to be my response, is when we've handled that, because you just assume, especially in reef environments, that there's high turnover, and so the persistence of any particular species can be fairly variable. There's some that are persistent, but a lot of them are, blink on and off for particular reef zones. So we address that by having multiple temporal samples for similar locations, just like you mentioned, Rick.
All right, well, with that, let's thank our speaker. Yeah, thanks so much, Erik. And then if we encourage everyone to stick around, you can turn on your videos now and yeah, chat.